Well, we will get started, David. First, I'd just like to begin by acknowledging that this webinar is being recorded on Mi'kma'ki, the traditional territory of the Mi'kmaq people. We are here with David Burton. It is our third webinar of the amendment series, our summer series. So, David, would you mind if I read your bio for
Oh, go ahead.
your crowd? Okay,
Keep
awesome.
it short, though.
Keep it short. There's Gordon's. Okay, so Dr. David Burton is a soil scientist and director of Dalhousie Center for Sustainable Soil Management. His research focuses on soil nitrogen cycle in the context of soil fertility and environmental impact. His current research examines the production and consumption of greenhouse gases in natural and agricultural landscapes, the development of tools for the measurement of soil nitrogen supply to plants, the influence of climate on soil biological processes, and the assessment of soil health. It is the aim of this work to better understand the factors that control soil microbial processes and to use this information to develop sustainable land management systems in a changing climate. So with that, David, you can get started and share your screen. And then I'm just going to turn it around.
So okay, well, thank you for inviting me to participate in this webinar. Adding organic amendments to soil is a very important process, but understanding what happens to those amendments and how soil conditions influence their behavior is really kind of important, and it's something I've spent a lot of time thinking about. So that's what I want to talk about today. So I'm going to share my screen. Okay, and so I've entitled my talk today, Converting Soil Organic Matter into Plant Nutrients, the Role of Soil Biology. So, like I say, I want to talk about what the organisms do and what either encourages them to do it or inhibits them, and how we can actually use that to inform some of our management decisions. Okay, so there are basically 5 things I want to talk about. First of all, I want to spend just a few minutes saying what's organic matter, or what's soil organic matter, and how is it formed. I want to talk a little about where plant nutrients come from in terms of that organic matter, and why organic matter sometimes releases nutrients, sometimes it doesn't. Then I want to move in and talk a little bit about soil health, the concept of soil health, and how that reflects the biological capacity of the soil and how we can use that information. And then I'm going to choose a specific example from our research, and that's a tool or a test we've developed to measure how much nitrogen is mineralized from the soil as an input to understanding our nitrogen budgeting process. And I think that's a really important way we can quantify and explicitly consider that impact of organic matter management in terms of plant nutrient supply. And finally, I want to just touch on some interesting work we've done on how organic amendments can increase soil organic matter and nitrogen mineralization. So let me get started on that. Okay, what's organic matter? Many of you probably understand what organic matter is, but I just want to really convey what I think organic matter is. So it's the carbonaceous material that's in the soil. We understand what organic molecules are. They contain carbon, oxygen, hydrogen, and nitrogen. But they play a really dual role in soil. One is they're the backbone of the soil. They're the glue that sticks all the particles together that creates soil structure. And that soil structure is what allows soil to function in the way that it does, hold water, resist erosion, and those sorts of things. So there's a really important structural role of organic matter. The second role it plays is it's the substrate for a diverse microbial community, not even just microbial, community in general, biological community. And that soil organic matter not only sustains that community, but it's the product of that community. And so it reflects this really interesting ecological zone, one of the most diverse habitats on our planet. And that diversity imparts really important function to soil. And one of those functions being the supply of plant nutrients, which I want to talk a little bit about today. One of the things that's really important to understand in agriculture, particularly in annual cropping, is where does organic matter come from and how does cropping influence that? Because too often we sort of think soil organic matter is really only a function of the organic amendment we had or the above ground crop residue. But in truth, it's primarily related to plant roots. Plant roots are the major source of organic matter. Here I've got a depiction of a corn growing, and if we look at 100% of the net primary productivity of that corn, 45% goes off in grain yield. 47% is the above ground biomass. This is the corn grain crops where we're leaving a lot of biomass behind rather than harvesting as a silage. If we're harvesting a silage, this would be more like 90% being removed. And then we have only about 8% that is remaining in the soil and the roots. However, if we look here at the formation of soil organic matter, those roots have a disproportionate influence on how much becomes soil organic matter. Why? Because if they're left in the soil in contact with the soil particles, with the soil aggregates, they have a potential to form that more stable soil organic matter. And that's something that's really important to realize. So soil organic matter is primarily formed from roots, ideally roots that are left undisturbed. So I want to leave you with that thought, and we're going to come back to that thought. So traditionally, when we talked about soil organic matter, we had all these theories about this as being this incredibly big, complex molecule that was associated with clays and couldn't be degraded because it was so complex. But now we've kind of updated those theories to realize that, yeah, that's part of it. So over here, we've got this association of various sizes of substance from various sizes of organics from the actual crop and animal residues that are added to the soil, like our organic amendments or our crop residues, the biopolymers that are the large biopolymers that are insoluble but are formed from those primary materials we've added, then smaller biopolymers that become soluble, and that solubilization process is really important, and they break down eventually to monomers and are decomposed to CO2. So we have this flow of energy and carbon through the system, but there's the opportunity to stabilize that material, either by association with mineral surfaces in something that looks kind of like this, or the newer way we're understanding it is the temporary isolation of that material into soil aggregates. So even though some of these small monomers might be very dynamic and easily degraded, they can get trapped within these soil aggregates and become organic matter for extended periods of time. It's really important for us to understand that potential as well, because that's when I'm going to continually talk about the issue of soil disturbance. And that's one of the ways in which soil disturbance can reduce the amount of organic matter that is in our soil. Okay. So I've introduced the concept of organic matter in a very, very, very quickly and briefly. I want to link that to the concept of the supply, the role of organic matter, organic amendments in terms of supplying nutrients. I'm going to come back to this previous diagram. This is mostly about carbon flow, carbon and energy flow. So as these big molecules are broken down, the organisms are breaking them down primarily to get energy. That's their food. So they're breaking it down to get energy and releasing CO2. But there was nutrients associated with these residues, primarily nitrogen, phosphorus, and sulfur. Not potassium, not calcium, not magnesium in nearly the same sense, but primarily nitrogen, phosphorus, and sulfur. And really what happens is when these organisms are processing these molecules, they take what they need to make more organism, but what's left over, they excrete as a waste product. And that's what we call nutrient mineralization. That's the nutrients that are become available to the plant. So really it's bacteria poop that we're relying upon to feed our plants. And that's really important. So this waste product, what we refer to as mineralization, is the soil nutrient supply that we're talking about. So one of the things I'm often asked is, well, when will mineralization occur? And that depends on a number of things. I think Gordon's talked a little bit earlier in the series about the role of the carbon to nutrient ratio. And in this example, I would use the carbon to nitrogen ratio. And really what that ratio is about is how much decomposition has to occur before that ratio narrows to about 20 to 30 to 1. 20 to 30 to 1 is where the organisms have enough nutrients to make new organisms. And anything when the ratio is more narrow than that, they excrete it out as waste products, as I suggest. So we'll get mineralization from organic matters that have C/N ratios of less than 20:1, things like soil organic matter, sewage sludge, alfalfa residues, farmyard manures, at least some farmyard manures, whereas things like corn stover, tree litter and tree bark, and conifer wood, they have very wide C/N ratios, and therefore they will not release nutrients. They'll actually grab nutrients so that because the bacteria need additional nutrients to make their bodies. So that's really important to remember, because when someone says, how long before I'm going to get mineralization of this organic amendment, it depends on the C/N ratio of that amendment. The other thing it depends upon is the activity of the microbial biomass. And that's largely about how we're managing the soil and the health of the soil. It involves things like the nature of the microbial community, the temperature of the soil, the moisture content of the soil. And that's where we come into introduce the concept of soil health. And I'm going to go there, but I want to also emphasize one aspect of this nitrogen supplying capacity of soil organic matter that is often misunderstood. So when we add organic amendments, we're contributing that to the soil organic nitrogen pool. And this red arrow is this process of mineralization that turns it into a plant available nutrient. I should also note that amino acids are also plant available, but too often they're catabolized or eaten up by the bacteria before they can get to the plant. So most often plants see ammonium, and that's nitrified. Again, organisms taking energy out of that ammonium to produce nitrate. And usually it's nitrate that accumulates in most of our aerobic soils because it's the form of nitrogen that has the least amount of energy. The bacteria have taken all the energy they can out of it, and they said, okay, let's leave the nitrate accumulate because we're not interested in it anymore. But what's really important is if we look at what the total amount of nutrient that's taken up by the plant, 50% comes from soil organic matter, and can be organic amendments can contribute to that, but 50% becomes from soil organic matter. Only 50% comes from the fertilizer N or recently added organic amendments. Okay, so we need to remember that soil mineralization, the mineralization of soil organic matter, is a really important source of nitrogen that's already there in our plant system. So when we're planning our fertilizer strategy and our nitrogen budget, we need to remember that there's an important and large source of nitrogen already present in our soils. So we've estimated, and I'll talk a little bit more about this later, that about 100 kilograms of nitrogen per hectare are being mineralized from soils in Nova Scotia each growing season. That represents about 0.2% of the soil organic matter and about 2.5% of the total nitrogen in the soil. That's a substantial amount of nitrogen, and we shouldn't be forgetting about it. I'll come back to that in a moment. How do I know that? I'll come back to that. Now I want to spend a little bit of time talking about how soil health influences that soil nutrient supply. Well, what do I mean by soil health? Well, we need to reflect upon the fact that soil does a whole lot of functions for us. There's a lot of things that soil does, and many of them are mediated by the organisms that live in the soil. And so when we talk about soil health, what we're really talking about is the viability and the diversity of those soil organisms, but in the context of their ability to perform these functions. So soil health is really about the ability of the system to function, much like our health is. And in fact, we can talk about various health in various contexts. So here I've presented a diagram where I have soil health in here, but we could also be talking about plant health, the health of the production system or the farm, the health of the ecosystem. And all of these things are impacted by different factors, some of them being biophysical, like the nature of the resource or the climate, but some of them also being related to our management, things like human capital, input costs, cultural expectations, and whatnot. These all impact health of various elements of the system. And so when I talk about soil health, I want you to remember that it's one sort of health that we need to be thinking about when we talk about the overall function of this system. But health is really about the system's ability to function. So I'm talking about the ability of the soil to function when I talk about soil health. So in terms of sustaining soil health or soil function, it's important that we think about how we can actually measure this. We need to assure that we have ways in which we can assess and measure soil health. And over the last decade, we've developed these measures, and they gave us important insight into how soil management, cropping systems influence soil health. And in fact, if we want to use the term sustainable, sustainability is really a response of management to this current condition of the environment, of the resource that we're managing. For a practice to be sustainable, it has to sense the resource that it's managing. And this is how we sense the function of the soil, by measuring soil health. There's a number of different aspects of soil health we can talk about. The physical things like structural stability, the stability of aggregates, the compaction of that soil, so its physical structure, its chemical structure, which is we've traditionally measured in soil tests, things like pH, buffering capacity, cation exchange capacity, the amount of nutrients chemically contained in the soil, but also biologically, the biodiversity of that soil, and that ability of that soil's microbial community to function, to respire, to release nutrients. That's part of what I'm calling soil health. One of the things we need to be aware of in Atlantic Canada is that we've had decades of declining soil health, and this is most evidenced by the decline in organic carbon content of our soils. And I know this map's a little hard to see, I apologize for that, but basically the darker the color, the darker the green the color, the worse the condition, the faster organic carbon is declining. And you'll see there's a lot of regions that are dark green, almost all of PEI, major sections of, you can't see the valley here because of the name Halifax, unfortunately, my apologies, but those dark green areas are where organic carbon is declining, largely as a result of annual cropping systems, the movement from perennial cropping systems into annual cropping systems. That's part of what I want to talk a little bit about. As part of this observation, we formed about 5 years ago, we formed the Atlantic Soil Health Lab. Now it's become part of the Center for Sustainable Soil Management. But as part of forming that Atlantic Soil Health Lab, we did a survey of the health of the soils in Nova Scotia and Prince Edward Island to try to say, let's measure this. Let's find out what the status of health is. We measured about 13 or 14 parameters, physical, chemical, and biological parameters. I'm just going to touch on a couple of them just for the interests of time. But I think there are a couple that really reflect the overall health of the soil and are very good indicators of soil health. The first is total organic carbon or organic matter content. You often hear the words organic matter content and organic carbon used interchangeably. Organic matter content refers to all the molecules in the organic molecule, the hydrogen, the oxygen, the carbon, the nitrogen, phosphorus, sulfur, whereas total organic carbon only talks about the carbon content. But carbon content is the main element in organic matter. So they're somewhat synonymous terms, although the numbers are different, the actual magnitudes are different. But I'm going to talk about the total organic carbon content. And what I've plotted here is the organic carbon content as influenced by cropping system in Nova Scotia. As influenced by cropping system. So what this is saying is the crop we grow and the way we choose to manage that crop has very important influence on the amount of organic carbon that is retained in the soil. And we notice a couple of things. First of all, look at the range in values even within a cropping system. Huge ranges. That means there's high variation in soil health. So this is an important thing we can measure, so we can work with people who are down here to say, how can I improve my soil health? But also we can work with the people who are up here to say, how do we make sure you're taking full advantage of the health and the improved function of your system? And do you understand how you got there so you can maintain that health? Okay, so being able to measure that is really important. But also it's really important for us to acknowledge the fact that these cropping systems have different ranges. In general, one of the things you will notice is that cropping systems that have a high degree of soil disturbance tend to have lower organic carbon contents. Things like field crops, and vegetable crops have the lowest organic carbon content, whereas things like forage crops or pasture systems or blueberry system, which is a perennial crop that has almost no soil disturbance, have much higher organic carbon content. Okay, I want you to keep that. Remember that concept of soil disturbance. Okay, so that's one of the messages I want to leave with you. But how does that relate to soil function? Well, here's another one of the soil health parameters we measured. This is aggregate stability. What are aggregates? Well, remember that that's the association of mineral particles and organic matter into little clumps or masses that define the physical structure of the soil. They're really important in terms of soil function. So here what we've done is we've measured the resistance of these aggregates to rainfall with the rainfall simulator. So 0.9 means 90% of the aggregates were resistant to rainfall. 0.1 means only 10% were. So in a healthy soil, we have aggregates that are resistant to the impact of rainfall, so they can hold themselves together, they can resist erosion. And again, we're looking at the effect of cropping system within Nova Scotia, and we see some interesting patterns. One, within field crops, we have a range. We have some of those systems that can maintain relatively high aggregates to water stable aggregates, whereas we have some that are quite low. These ones are going to function much more poorly. They're going to be more droughty. They're going to be have more potential for erosion. Forage systems, we see two peaks. We see some forage systems don't have very high aggregate stability, where some have very high. Why? What's the difference? Are these two forage systems being managed such that one does maintain aggregates, one does not? Pasture systems almost invariably have very high aggregate stability. Why? Because they often involve perennial crops or periods of time when the crops are left, the soil is left undisturbed for a period of time. Vegetable production systems, very low aggregate stability. Why? High degree of disturbance. This is a system where we're going in and we're manipulating that soil on a relatively frequent basis, and that is impacting the amount of carbon that's stored in that soil, but it's also impacting the stability of those aggregates and impacting the function. And again, this perennial wild blueberry system, very high aggregate stability, water stable aggregates. Again, a healthier system because there's more organic matter and more water stable aggregates in that system. Okay, now I want to take another specific look at soil health and talk specifically about one of the processes that's near and dear to my heart, soil nitrogen and soil nitrogen mineralization. So one of the things we did as part of the work we did under the Soil Health Lab is to say, is there a way we can actually measure the amount of nitrogen mineralization that's occurring in our soils? One of the problems we've had in Nova Scotia is that we don't have a soil nitrogen test. So many of the times when producers are trying to make a nitrogen recommendation, they don't really have anything that's measured about their soil that they can use to inform that. And in particular, while in some parts of Canada, we can measure the amount of nitrogen that's going to be carried over from the previous year, in Nova Scotia, almost no nitrogen is carried over. So it's really mineralization in the growing season is the number we want to know, but we didn't have a test for that. Well, we do now. That's one of the things we developed. So working over the past 20 years or so with colleagues with Agriculture and Agri-Food Canada, like Bernie Barath and Juvius Niza and Keith Fuller, we worked on developing what we call a biological nitrogen availability test. And I'll explain that test to you in a moment. But what it tries to do is it tries to measure the amount of mineralization that would come primarily from soil organic matter, but from the long-term return of crop residue and the long-term use of animal manure. And I say long-term because we're not so sure this test is as effective in reflecting the contributions of recent additions of manure. And that's something we're continuing to try to work on. But it gives us a way of quickly and easily measuring the nitrogen mineralization potential of that test. Okay, and I'll get to this in a second, this next part. But what does that test look like? Well, it's a super simple test. What we do is we take a soil, we air dry it. That's important. We air dry it. Then what we do is bring it to the lab, re-wet it, and that re-wetting causes a burst in microbial activity. A flush of microbial activity, but we also, when we're rewetting it, we leach out all the ammonium and nitrate that was already contained in the soil. And that's what we call the residual soil nitrogen. That's what we would have normally measured in a soil nitrogen test. That's the only part. We then incubate it for 2 weeks in the laboratory and we leach it again. So we measure all the ammonium and nitrate that was formed by the bacterial community or the microbial community during that 2-week period. And that's what we call biological nitrogen availability, or BNA. That's our test. It measures the potential of that soil to mineralize nitrogen. Back to this diagram. So we now take that measurement of mineralization potential, and then we put it in the context of the climatic environment and the pedological environment of that soil, and say, how would climate influence the degree to which that potential is realized? And we get an estimate of growing season nitrogen mineralization. That's the information I want to present to you. And I want to answer the question, how big is that potential? And how does it influence by things like cropping system and the addition of organic amendments, things like that? So again, going back to our soil health survey, here is the growing season nitrogen mineralization that we observed in all of those soils we sampled under various cropping systems. So it's a survey of about 800 fields, which is a fairly big number, not as big as we'd like, but it's fairly significant. A couple things I want you to notice. Look at the extreme variability from hundreds of kilograms of nitrogen per hectare to tens of kilograms of nitrogen per hectare. Right now, we're treating those soils as if they were all the same in terms of our nitrogen fertilizer management. We're not reducing nitrogen fertilizer up here. We're not adding more nitrogen fertilizer down here. We're considering them all the same. So this is really important information that we've been missing. Now we have a way of getting at that information. So that incredible variability. Also notice some of the trends where we're seeing some of the highest values. Forages, where we include legumes often, pasture systems where there's legumes, and also systems where there was higher organic matter, we're seeing higher levels. Where we're seeing lower levels are these high disturbance systems, field crops and vegetable systems. Again, part of that disturbance is the fact that organic matter is depleted, it's supplying less nitrogen. We also did this work in Prince Edward Island, where we have a bit more of a larger data set, about twice as many samples, seeing exactly the same sort of trends. Forage hay systems have much higher potential growing season nitrogen mineralization than do, for example, potato systems or field crops. It's important that cropping system does influence this potential. But again, extremely high variation from field to field, variation that we're not capturing. And notice the magnitude of these numbers. We're talking about 100, 150 kilograms of nitrogen being mineralized per growing season. We've also tried to make this number more accessible. This is the work of one of my graduate students, Luke Lawrence, who just defended his PhD. And so what he's done is he's generated what we call a pedo transfer function, or an estimation function, to generate a map of growing season nitrogen mineralization based on soil characteristics for the whole province of Prince Edward Island. So a producer in Prince Edward Island can simply locate their farm to get a general idea of what their nitrogen mineralization rate during the growing season should be. So that's a really very good first start, but it's only an approximation. So the other thing that Luke did was create a way in which we can use these approximations to actually generate a nitrogen credit that we can use in terms of developing our nitrogen fertilizer management system. So here he's talking about 3 tiers. Tier 1 is when I just picked it off the map. I didn't have any soil test at all. I just picked it off the map. So this map, I found my farm and I said, okay, the number for my farm is 134 kilograms of N per hectare. But Luke said, you know what? We have some uncertainty in that number. I'm a little uncomfortable telling the producer that they can rely upon that. So I used the machine learner that generated this pedo transfer function to say, how certain are you? And that certainty came back at 60%. So we'll only take 60% of this number. We said 130-day growing season, but for the crop I'm growing, there's only so many days of that growing season that's really actively taking up nitrogen. So we're only going to take 70% of that number to reflect the active period of plant uptake. And we know that nitrogen applied to soil, whether it be fertilizer or from soil organic matter, is only 50% efficient. So we take our 134 estimate and come up with an estimate of about 28 kilograms of N per hectare. We feel much more comfortable in terms of telling the producer that that's the amount of nitrogen they can count on. However, we've got some producers that said, well, maybe I didn't do the full biological soil health test to get a measure of BNA, but I did do the standard soil where I measured the standard nutrient regime. Well, what Luke found is if he had organic matter, pH, and CEC, he could estimate that nitrogen mineralization potential with somewhat higher confidence. So we only reduced that estimate by 70% rather than 60%. But we applied again this 0.7 reduction factor for the reduced growing season and the 50% efficient. And now our estimate is 32 kilograms of N per hectare. However, this particular producer actually did do the PEI soil health test, which includes both the measurement of total nitrogen and the biological nitrogen availability. So we can actually put those values in from those fields, and found out rather than an estimate of 134, 132, this field wasn't quite as good as that typical provincial average. It only had an estimate of 102 kilograms per hectare. But we have greater confidence in that estimate, so we can apply a risk factor of 80% rather than 60 or 70%. But again, the 0.7 and 0.5. So now our nitrogen credit is 29 kilograms per hectare. So this is an example of how we can use our understanding of that soil biology, its effect on total nitrogen, to get a really good estimate of how much nitrogen supplying capacity our soils have, and monitor that over time. Okay. Again, I think it makes it more tangible or more quantifiable about how our management is affecting our capacity of soil to produce. Final thing I want to talk about is another interesting observation we made as a result of that survey of soil health in Nova Scotia. It has to do with the role of organic amendments. So we had about 800 fields that we sampled. Some of them had added amendments. Some of them had not. Some of them were in high disturbance systems, and some of them were not. So we grouped the soils that were amended, and we grouped the soils that were non-amended, didn't receive organic amendments, and partitioned them into ones that had high disturbance versus ones that had low disturbance. And one of the interesting things we saw is total carbon accumulation was greater in amended systems that had low disturbance, or even in non-amended systems that had lower disturbance, and was much higher certainly than non-amended soils with high disturbance, but even higher in some cases than the amended soil with high disturbance. So it appeared that the degree of disturbance was far more important in determining whether we're building up our soil organic matter than simply was the addition of an amendment. And that's really important because sometimes when we're adding amendments, we're doing a lot of tillage to incorporate that. And sometimes we have to think, are there ways we can put that amendment in a way that it's more stable so we won't get losses, but we can actually do it without disturbing the soil very much. What about nitrogen? Well, not quite as dramatic a picture, but we found a similar sort of thing. The amended high disturbance systems did not necessarily mineralize as much nitrogen as the amended low disturbance systems or the non-amended low disturbance systems. Why? Because again, we're building up that longer term organic matter. And I highlighted that we're not clear whether our BNA test is doing a very effective job in reflecting recent amendments. So these soils were amended. There were soils that had been amended over the long term, over a decade or so, had continually received amendment, where organic matter would be anticipated to build up. Again, the message I want to leave with you a little bit is adding organic amendments is really important, but it's also important how we manage those amendments. And too much soil disturbance can negate the effect of that amendment, both in terms of building up organic matter or enhancing the soil function, the soil health and soil function of that soil in processes like nitrogen mineralization. Okay, so those are the things I wanted to talk about. I wanted to talk about what is organic matter, how is it formed, and I want to really emphasize the fact that part of it is how complex that organic matter is and how it associates with things like clay minerals, but also part of it is how much it's disturbed. how much we can just leave it alone and let the organic matter form. Where do the plant nutrients come from? Well, the decomposition of that organic matter. Soil health influences that by influencing the activity of the microbial population, but also influencing the structure of the soil, the formation of those aggregates that will allow the soil organic matter to form. Again, this influence of tillage and disturbance on allowing that formation to occur. And when I just demonstrated how we could show that within nitrogen mineralization, and how now we have a test that we can actually measure the nitrogen mineralization potential during the growing season in our soils in Nova Scotia. I should say that the BNA test is now part of the standard package that the PEI Analytical Lab is offering to its, not the standard package, sorry, the soil health package, but the PEI lab is offering to its growers. And now the Nova Scotia lab is also developing BNA as a test that will be commercially available to growers in Nova Scotia. So that's tremendous progress. And finally, I ended with this example of organic amendments where I said, basically, it's not just the addition of the amendment, it's also how much we disturb the soil once we've added that amendment. It's really important in understanding how much we build organic matter and build soil health. So I'm going to end there, but I want to leave you also with one other piece. I've been working with a group called Farmers for Climate Solutions, and one of the things they've been doing is developing what's called the Farm Resilience Mentorship Program. And this is a free program to all producers in Canada that highlights a lot of the kinds of practices that are part of your off-calf program, things like nitrogen management, rotational grazing, cover crops, and offers the kind of guidance and information that I presented today in a way that you can observe it at your own leisure. There's also, because it's a mentorship program, there are mentors, so you can actually talk to farmers who are implementing these practices and get some more direct advice from them. So I just wanted to end by sort of making you aware of that program. Again, it's a free program. All you need to do is go to the farmlearninghub.ca, sign up, and you have full access to the site. So with that, I will stop sharing my screen and see if there are any questions.
Thank you so much, David. That was fantastic. Really appreciate that. I have some questions, if and then we can wait until some others come in. Your last slides there, looking at total carbon and nitrogen as well, I think that really hit home. For me, I think my mind just automatically goes to amendments, and that the amendments would therefore increase nitrogen, carbon, so on and so forth. But I don't know, it kind of really shifts the gears in my mind personally, when in how undisturbed soils, that's what increases the total nitrogen and total carbon. So yeah, that it just hit home a lot.
Well, the analogy I often use is it's like pouring gasoline on a fire. So if you just pour it straight on, if you just add the organic amendment, you're just going to speed up the whole decomposition process, which will release a lot of nutrients in the short term. So that may be your objective, and that's good. That's fine. I'm criticizing that. But I'm saying if your objective is to build up soil organic matter, store that gasoline, you have to handle it a little bit more carefully.
Yeah, exactly. Okay. We have one question here. What is your opinion on those soil nutrient sensors that can be used in field? Are they reliable?
Well, I'm not sure which soil nutrient sensors you are referring to. We use some what are referred to as ion exchange membranes, which is a commercial technology out of Saskatchewan called the PRS, or Plant Root Simulator Probe. And we find that they're really quite useful, because what they're doing is, for example, we use anion exchange probes which absorb nitrate. So they actually tell us how much nitrate is becoming available in that soil and under the conditions of that soil. So under things like wetting and drying, different temperatures. So if that's the sensors you're referring to, we find them quite useful as research tools. They're a bit expensive for an on-farm tool, but I think they're quite reliable. But I'm not sure if that's the sensor that the question was referring to.
Not sure. That's also helpful, though.
There's also sensors of the crop, like the green crop, like leaf sensors. Those measure chlorophyll, and again, they can be quite reliable.
They say they've just started looking into them, so they're not quite sure, but just looking for
Okay.
information there.
I really like the idea of sensors that allow us to measure things. I think one of the other major messages I want to get across is we have to measure more things about the state of our soil and its function. Too often we've just assumed, well, the soil. I'm going to really worry about the crop. I need to worry about the soil too.
And do you have much experience working with agronomists or larger organizations, maybe out west, that are doing that for farmers?
Yeah, there are a number of organizations that are trying to do that. In part, sometimes it's associated with a nitrous oxide emission reduction protocol. There's some groups in Alberta that are trying to do some of this monitoring. It's not really cost effective at this point. I think as the technology develops more and becomes more available, it might become more practical in terms of its cost. And also, we don't necessarily have the agronomy to interpret it all that well. So it's very much emerging technology. So that's one of the reasons why Luke did the work that he did in using pedotransfer functions, because one of the philosophies behind what he was doing is, can I give the farmer information based on the data they have? Rather than saying, gee, if you bought all this additional stuff, or if you had all this additional information, I could tell you something. But based on the data that you have, even if it's just the location of your farm, can I give you at least something? And I think we're going to see, as big data emerges in our analytics and understanding the nature of soil and whatnot. I think we're going to be able to give producers more information, even if they don't have a lot of sophisticated technology in their field, because soil is a very complex, self-regulating body. But we can learn some things about that self-regulation, and we can use that self-regulation as a way of narrowing down the options, if we will, or better predicting how it's going to behave in terms of response to temperature and climate. Those things.
It was interesting too with the different tiers that Luke provided. There wasn't a huge, range. They were fairly similar in terms of what was available for nitrogen.
Yeah, they weren't crazy different. It was interesting that the actual one that was based on measurement was lower. But then when we went back and thought about that was an experiment that the East Prince people did as part of their original living labs. And we realized that when we asked those producers to sample their fields, we asked them to sample their worst field, because we wanted to see, could we document impaired health? And yeah, compared to the provincial average, those soils were mineralizing less. And the BNA test was able to detect that they were mineralizing less.
Very cool. Very cool. So that's going to be available for producers within Atlantic Canada?
Well, it is already available in Prince Edward Island. So you can send samples to the PEI lab. And I think New Brunswick are already sending some of their samples to the PEI lab. It's not an inexpensive test. But the Nova Scotia lab is also developing the BNA test as a service that they're going to provide. So I don't know, time frame, a year or so, I don't know. We're working with them to do that. So we're doing it as quickly as we can, but I think it's going to be here soon, for sure.
That's really exciting. So with that too, if producers were to do that sample, would they take a sample pre-amended soil or post, or would it matter?
Again, we were trying to be super practical when we developed this, and we said producers don't really have the time in the spring, and we don't have the time to process the sample. So what we said, sample the fall before.
Okay.
We know scientifically that wouldn't be the optimal time, but can we show that organic nitrogen supplying potential is stable enough that if we sample it the fall before, it's still reflective of what's going to happen in the next growing season? And it turns out it is. It doesn't vary very much from year to year, because it's based on, in part, the total nitrogen content of the soil, which doesn't change very much. So we actually suggest that producers sample after they harvest in the fall. Again, air dry the sample and send it to the lab. So we've got some time or the lab that's doing it has some time to process that sample, get the results, send it back to the producer to inform next year's nitrogen management strategy, right? Again, trying to be super practical. So this is a usable test.
Very exciting. So, pumped. One question here. Do you know?
I realized I didn't answer your question though.
Oh, that's okay.
Wait, no, it's important. One of the things we're not clear on is if you did sample right after you added an organic amendment, I think we're not going to get a good sense of the nitrogen supplying capacity of that recent organic amendment. Or similarly, if you just recently plowed down a nitrogen-rich cover crop.
Okay.
The test is probably not doing that very effectively. So one of the things we're looking at is, do we still need to generate a manure credit or an organic amendment credit and a legume credit separate from the soil mineralization credit? So that's an area of uncertainty that we're not sure. I think some of the results I showed you in the last two slides may have also reflected that we weren't necessarily capturing the nitrogen supplying capacity of those high amendment systems as effectively. So I don't know is the right answer to that question.
Either way, it's another tool, right? You can send in your
Yeah.
field history, crop history, do a sample, and you're gonna get some good information.
Well, and one of the things we really want to do is give the producers as much information about, again, my focus has been nitrogen, but we can talk a little bit in the same way about phosphorus. Giving them as much information to give them an understanding how much nitrogen is available in their system so they can add as little fertilizer as needed. Because adding too much fertilizer is neither economic or environmental. So if we can give them confidence that, no, I can count on this amount of nitrogen being available, I only now need to add this additional amount, that works out well for everyone.
Yeah, it's peace of mind, right?
Yeah, but we don't want producers to have crop failures. So that's part of the reason why Luke developed those reduction factors to say if we're not so certain, we're going to be conservative.
Cool. Do you know if there has been any research done in terms of the use of multi-spec drones in soil monitoring?
There has been some. So not too often. Most often, the drones are measuring the crop, not the soil. There has been some work in PEI where they're actually using drones and also satellite imagery to measure bare soil and trying to infer some things about the state of bare soil from again, both, I guess initially it was drone imagery, but I think in many cases satellite proved to be just as effective because satellites are becoming amazingly more powerful than they used to be, and multispectral as well. One of the really cool things that we're trying to do as part of the center is we're trying to use a multispectral analysis within our lab to do a spectral analysis of soil that can predict a wide range of soil properties, from the traditional chemical indices we measured in the soil test lab to things like organic carbon, nitrogen mineralization potential, those kinds of things. Because if we could get a spectral analysis, one analysis that gave us a large data set, a wide information or a broad amount of information about the soil, that would be really useful. And so we're primarily looking at that as a laboratory tool. Maybe in future that could also be an in-field tool. I don't know. But I do know there is at least some work looking at multispectral analysis of soils using drones and satellite information.
Cool. I would like to go back to the carbon and nitrogen ratio and a bit of soil health and nitrogen amendments. And I think I get overwhelmed, thinking about this stuff. But when it comes to nitrous oxide emissions after application, how does the carbon and nitrogen ratio of those amendments or those soil health parameters either increase or decrease maybe some nitrous oxide emissions?
Yeah. Well, that's an important point too, because nitrous oxide is actually something the bacteria do as part of their respiratory system. So when they're respiring a lot, they have a high demand for terminal electron acceptors, sorry for the technical term, but things like oxygen and the things that cause nitrous oxide to be formed. So one of the things we do see is that in manures, because they have so much carbon, they really stimulate the microbial population. There's a lot of water there, so often oxygen supply is limited. So that's when we can get significant denitrification and nitrous oxide emissions. And so, or for example, in a solid manure stack, there's enough carbon there that there's intense microbial activity. It's wet enough that you're getting limited oxygen, and therefore you can get significant N2O emissions. Interestingly, in liquid manure systems, we don't see that because there's almost no nitrate forming. It's so anaerobic that most of the nitrogen stays as ammonium. So we don't get as much nitrous oxide from a liquid manure system or a liquid manure injected into the soil than we would, for example, a solid manure. It's clearly left in the soil or on the soil surface, but even incorporated in the soil surface, and that's why it's a very, like I'm saying, oh gee, we should put our organic amendments on and not disturb the soil. But if we leave it on the soil surface, there's a potential for nitrogen loss. So I'm not sure I have the right answer yet, but we need to think about that balance of minimizing disturbance while at the same time putting the amendment in the soil where it can do good. And that's, like, for example, the injection of liquid manure. I think that's an excellent example of where there's not so much soil disturbance, but we're putting the manure right down in the soil where the ammonium is retained by the soil. The alkalinity of that manure is neutralized, so there's less potential for ammonia loss. There's less potential for nitrous oxide loss too. And so that's a more ideal situation. Solid manure is tougher. Composting, maybe that's the answer. You'll like that answer.
Yeah, that's awesome. And I guess timing too, that's another factor that I guess we have to consider, but
yeah, timing's important as well.
David, that might be it for questions.
Okay, cool. Well, this has been fun. I hope it was useful.
It was very useful, and I really appreciate your time today. So thank you so very much. And it will be recorded, so this will be on the OffCap website forever.
Okay, excellent. I don't know if that's a good or a bad thing, but there you go.
Oh, thank you so much.
Okay, take care.
Yeah, you too. Bye.
Bye.